Hello, everybody. Welcome to Ms. McGuire online lectures. And today we are covering the cellular level of organization. So let's go ahead and begin. So first we want to start with what it's called in biology cell theory. And the cell theory states that cell is a structural and functional unit of life. It also says that all cells come from pre-existing cells, and all living organisms are made of cells. So cell is the smallest unit of life. It's a structural functional unit of life. All cells come from pre-existing cells, and life on Earth has a cellular basis. So how well the entire organism function depends on individual and combined activities of all its cells. So pretty much anything that happens in your body would happen on a cellular level, right? So if your cells are not functioning properly, then your tissues are not working, then your organs are not working, and the whole organism is affected. Now, we just said that cells can arise only from other pre-existing cells. That means cells will divide to produce more cells. And you need cell division for if you want to replace some cells that can be damaged or lost. For example, if you cut yourself, right, some of those cells are dead. Now adjacent cells will divide and replace, stem cells, precursor cells, they will divide and replace cells that are damaged. You also need cell division for growth. When you were born, you were way smaller. So now all your organs or parts of your body are getting bigger. So most of this growth involves cell division. And we need cells for reproduction. We are sexually reproducing organism, so we make gametes, eggs, and sperm. So we will have some precursor cells that will divide, producing ova and sperm for reproduction, right? Now, structure and function are complementary. We cover it also in our first chapter, I believe. Now, that means that biochemical functions of cells are dictated by shape of cells and specific subcellular structures. Now, cells are surrounded by extracellular material. So extracellular matrix are substances found outside the cells, and it will include extracellular fluids. We also call them body fluids, such as interstitial fluid or so interstitial fluid or tissue fluid, another name. So cells are submerged or bathed in this fluid. Extracellular fluid also include plasma, that's a fluid of blood, a cerebrospinal fluid that surround your nervous system. Also, extracellular material includes a cellular secretion such as saliva, mucus, oil, and extracellular matrix. Those are substances, they act as glue to hold cells together. Right, so pretty much your body is made of cells, and it's also made of extracellular material, such as fluids, secretion, and extracellular matrix. Now, cells, there are lots of different cells, right? So we have a high variety of cells, of animal cells. However, doesn't matter what cell is that, all cells have some common structures and functions. Now, human cells have three basic parts. Every cell will have plasma membrane, that is flexible outer boundary, cytoplasm, that includes all the fluid inside the cell that is called cytosol and all the organelles, not including nucleus. So it's a space. If you look over here on this diagram, cytoplasm would be everything that is located between nuclear envelope and cell membrane or plasma membrane. And of course, nucleus, when we're talking about generalized cell, and nucleus contains DNA and some RNA, right? But DNA probably, you know, the main component of nucleus, right? So again, we have plasma membrane or cell membrane, cytoplasm, and nucleus. Very few human cells, type of human cells, they might not have any nucleus, like red blood cells. When they are formed, they have nucleus, and then they get rid of it, right? But most of the cells, they do have nucleus. So we will cover a major organelles and structures of a typical cell, and we're going to start with cell membrane. So cell membrane is made of several molecules, but mostly it's made of phospholipids. Now here you can see diagram of a cell membrane. It's a bilayer, and it's phospholipid bilayer. So cell membrane separates intracellular fluid from extracellular fluid. So cell, you know, animal cells especially, they must be surrounded by fluid, otherwise they will die. And cytoplasm is also mainly water. So we have fluid on both sides of this phospholipid bilayer. Now the phospholipid is shown over here. The diagram of a phospholipid molecule shows here. So it's a fat, and it's made of glycerol. So that's a glycerol molecule. Two fatty acids attached, and another group is attached, that is phosphate group. Now, where we have this glycerol and phosphate, we call it a head. And those fatty acids are called tails. Now, so we have one molecule that has two parts, and these two parts have different properties. Head is hydrophilic. Hydrophilic means it's attracted to water, and tails are hydrophobic, so they repel water. And because of this property of molecule, we call it amphipathic molecule. When those molecules are surrounded by fluids on both sides, they form this bilayer. Now look, because those tails, they are hydrophobic, so they hate water, they repel water, they will be oriented away from solutions, from watery solution, right? Aqueous solution. So here we have lots of water outside, lots of water inside. So those tails are oriented away from water. And heads are hydrophilic, so they love water. So they be facing the extracellular and intracellular fluids. So that's a very important property of phospholipids that allow them to form those biological membranes. So over here we can see another diagram that represent structure of cell membrane. And as you see over here, 75% are phospholipids that form lipid bilayer. And again, it repeats over here, phosphate heads, they're polar, and hydrophilic, and polar means they have a charge. And fatty acid tails, they're non-polar and hydrophobic. And on this diagram, you can see this phospholipids right over here and here and right there. 5% are glycolypids. Those are lipids with polar sugar groups on outer membrane surface. So if you see these branches of the molecules, those are oligosaccharides. So those are, sugars, but the chains are not very long. And they attach to these lipids. And when they attach to lipid, that's what we call glycolipid. And 20% of lipids are cholesterol. Now, cholesterol is inside this bilayer and increases membrane stability and fluidity. Right, so those are lipids. However, you can see what's shown here in this blue color, those represent proteins. So we have also different proteins within cell membrane, and they can be divided into two classes: integral proteins and peripheral proteins. Integral are firmly inserted into the membrane, and most of them are transmembrane. That means they go all the way through this layer. Peripheral proteins loosely attached to integral proteins. They can be inside the cell, they can be outside the cell as well. Now, so you see these blue structures are proteins. So this is our integral protein, and it's transmembrane. This is peripheral protein. But if you see this like a helix shape, this is also integral transmembrane protein. Now, glycocalyx is this sugar covering on the outer surface of cell membrane that will be shown better, I think, on some next slides over here. I don't know, not this slide, but maybe next one. So we continue with proteins, and we will get back to glycocalyx, okay? Now, what are the functions of those proteins? Now you see the function shown in these yellow boxes. Now, one of the functions shown over here, it's attachment to the cytoskeleton and extracellular matrix. So what you see here, you see two cells. Now what's shown in kind of yellow color, this is cytoplasm, so we're inside the cell. And whatever shown over here is the blue color, this is our extracellular matrix. Now, extracellular matrix includes fluids, so water and electrolytes, but it also has extracellular proteins. Now, whatever says over here, like fibers or extracellular matrix, those are structural proteins over here. And we also have proteins as a part of cytoplasm, so they make cytoskeleton of the cell. And these proteins, transmembrane proteins, what they do, they attach these cells through the cytoskeleton. So they attach to cytoskeleton inside and extracellular matrix outside, and pretty much anchor the cell. So allow it to stay in the same place and, not migrating all over your body, right? So another function is signal transduction. So very often your endocrine system or your nervous system gonna send special signaling molecules to your cells, because your nervous and endocrine systems, they want your cells to do something, to perform some function. Maybe they want the cell to start breaking glycogen and release glucose, or maybe they want the cells to divide. Right, so anyway, when cell receives those signals, some of these signaling molecules, they cannot move inside the cell. Instead, they will be attached to a receptor protein and, transduce or send the signal from outside to inside, and cell will respond. Another function is transport. Some molecules, they can diffuse across a cell membrane. Those are small non-polar molecules, some water molecules, even water considered to be polar molecule, but some water molecules can diffuse through. But many molecules like ions, sodium ion, potassium, calcium, or larger molecules like glucose or fructose, they cannot diffuse through the membrane, so they will have a special gates, we would say, or transport proteins. And some transport proteins, they are leakage proteins, so they always open and stuff is just moving down its gradient, or some proteins require energy to move stuff across a cell membrane. Another function is intercellular junction, right? So cells, some cells, you want them to be connected to each other. For example, your epithelial cells of your skin, you don't want gaps between the cells. So imagine if those are cells of your skin, you don't want this gap. You don't want water, you don't want bacteria, you don't want harmful substances move between the cells, or you don't want to lose water, you don't want to dehydrate if, for example, water moves in between cells. So you would have proteins that join those cells together. So junction. Another function is cell-to-cell recognition. So, for example, cell of your immune system need to recognize your own cell. So they will actually rely on this glycocalyx that we will discuss in couple minutes, right? But that's a cell-to-cell recognition. And this sugar is attached to a protein, making it glycoprotein. And another cell will have a special receptor that will recognize this glycoprotein. And one more function, enzymatic function. So some cells, some proteins, membrane proteins, can be enzymes, right? So they have this active site, and substrate can feed in this active site, and enzyme will initiate and speed up a chemical reaction. So you can see that proteins, membrane proteins, have very important functions, right? So again, attachment, signal transduction, transport, junction, cell-to-cell recognition, and enzymatic activity. Now, remember I told you we will discuss glycocalyx. Now, glycocalyx consists of sugars, carbohydrates, sticking out of cell surface. Some sugars are attached to lipids and making glycolipids, and some to proteins, making glycoproteins. Right, so if you see over here, we can see that this sugar attached to lipids, so now we can call this lipid glycolipid, and this sugar attached to protein, so that's a glycoprotein. Now, if you look at electron microscope here, you can see like glycocalyx on a surface of endothelial cells, and it really looks amazing. It's like the whole forest of these sugar molecules covering cell completely. And every cell type has different patterns of the sugar coating, functions as specific biological markers for cell-to-cell recognition, and your immune system recognizes self versus non-self. All right, so this is the glycocalyx on the surface of the cells. Now here's some clinical considerations over here of homeostatic imbalance. Glycocalyx of some cancer cells can change so rapidly that immune system cannot recognize the cell as being damaged. And so what it means, if your cell became a cancerous cell, and this is what happened, a cancer is actually, it's a disease of our own cells. Now, if cell became cancerous, you have immune system, those white blood cells, natural killer cells shown over here, that have the ability to recognize a cancerous cell and ability to destroy it. But process of recognition is very specific, very precise. So you can see over here, like some receptors of these natural killer cells need to be deactivated, some need to be activated. And, when everything fits, so here we have receptors, here we have glycocalyx of the cancerous cells. And if everything goes how it's supposed to, then natural killer cells can release enzymes that will destroy, or chemicals that will destroy cancerous cells. But because the cancerous cells can mutate, what they can do, they can deposit, it's shown here in the red color, they can deposit acid on these glycocalyx chains. And when they deposit those acids on glycocalyx, now your immune system now cannot recognize the cell. So for example, this receptor that need to be deactivated now activated, this kind of doesn't fit properly, this does not fit at all. And as a result, we cannot release those chemicals that would destroy cancerous cells. So mutated cell is not destroyed by the immune system, so it is able to replicate. And this is where we can have malignancy, right? And tumor will grow, and tumor can become malignant. So that's the importance of glycocalyx. All right, so we continue talking about membrane. Now, what did we discuss? We discussed structure of cell membrane. We said that this is phospholipid bilayer. But when we're talking about function, this is, the membrane is semi-permeable. And semi-permeable means some substances can go across the membrane and some cannot. And when substances, ions or molecules moving across a membrane, we call it transport. So we have two types of transport, active and passive. So here diagram, the small diagram here just represent that passive transport doesn't require ATP, and its analogy would be if you're on the top of the hill and you just let this ball go and it just go down the hill just because it go from the higher level to a lower, right? It will move because of the gravity. So you do not apply any additional energy. So that's passive transport. And active transport shown over here when you need to push this ball up. Now what this hill represents in biology, it represents a concentration gradient. So if we have high concentration gradient, then substances will move down its concentration gradient without any energy from ATP. So we don't need ATP energy to do it. Now, when substances moved from low concentration to high concentration, we say they move against its concentration gradient, and then it will require energy of ATP. Right, so on this diagram, you see examples of passive transport. So one example is simple diffusion. Now we have high concentration here, and we have low concentration. Right, and these molecules just move across the cell membrane without any help, without any ATP present, just moving down its concentration gradient, like shown over here. Now, what molecules would they be? It would be a small non-polar molecules. Now, if molecules are larger, or if they have a charge, for example, sodium, potassium, they're not that huge of molecules, but they are ions, so they cannot move through the membrane. Then they might use special channel proteins, right? And then we call it facilitated diffusion. So it doesn't matter, I mean, it's still diffusion because we're still moving from a high concentration to low concentration, but we use a help of membrane proteins. So it can be non-specific channel protein when, anything can pretty much move through it, or it can be specific channel protein when just one specific substance can move down its concentration gradient. And the last example is osmosis. Osmosis is diffusion of water. And water can move through cell membrane, but most of the water will move through channels. They always open. They call it aquaporins. So they move through these channels. But water also moves down water concentration gradient. So if you have lots of water over here and we have lots of salt here, that means we have less water, then water molecule will move where salt is, down water concentration gradient, right? So those are passive transport. Now, active transport require ATP energy. When we talk about active transport, we have primary transport and secondary active transport. Now, what is the difference? Now, so let's say this is transport 1, this is transport 2, right? Which one of them is primary, which one of them are secondary? Now, the primary transport directly uses ATP energy, and it moves substances against their concentration gradient. So example over here, sodium-potassium pump. What it does, it pumps sodium out of a cell and brings potassium in, and it uses ATP directly, right? Now, if we have, imagine if we have a chemical that will destroy this transport over here, the sodium-potassium pump would not be affected. It will continue doing its job. So we can destroy or block those purple proteins, and it will not affect our primary transport. Now, secondary transport, however, is right here. And secondary transport depends on the primary transport. And it does not use ATP directly. So now if we have chemicals that block the sodium-potassium pump, this transport shuts down as well. Now, why the secondary transport even works? So first of all, what we don't need to remember that secondary active transport moves substances against the concentration gradient as well. Now, what those purple or pink, pink proteins move against the concentration gradient? Actually, glucose. Now, this is what happened. Primary transport uses ATP and pumps sodium out. Now, if you constantly take sodium out of the cell, you have high concentration of sodium right there in extracellular fluid. You have lots of sodium. And now if you have proteins that allow sodium to return back, it will do it. So sodium will move through this pink protein down its concentration gradient, but sodium will take glucose with it, and it will move glucose against its concentration gradient, right? So we call it co-transport. And in this situation, it's a symport because sodium and glucose moves in the same direction. Sometimes we can have antiport when one ion, for example, moves in, or one molecule and another moves out. So secondary transport is co-transport. It doesn't directly use ATP, but it depends on the primary transport. And if primary transport stops working, that secondary transport stops working as well. Both of them move at least something against the concentration gradient. So here, sodium and potassium both are moved against concentration gradient. In this co-transport, sodium moves down its concentration gradient, but glucose moves against its concentration gradient. Right, so that's active transport. Another example of active transport would be endocytosis and exocytosis. Okay, so endocytosis is the movement of substances out of a cell in a form of vesicles. So some kind of vesicle need to be, oh, I'm sorry, did I say out? Inside the cell, Endo means inside. So let me repeat it again. Endocytosis is a movement of substances inside a cell with the help of vesicles. So here's 3 different types. We have receptor-mediated endocytosis. Now, what's shown here in yellow is inside a cell, cytoplasm, and blue is extracellular fluid. Now, if we have some target molecules, and target molecules are also called ligands, right here. So if we want these molecules inside the cell, those molecules first will attach to their receptors. So you see those are proteins, receptor proteins, and this will target the formation or induce the formation of vesicles. So vesicle will be formed only when those target molecules attach to the receptors, and these vesicles containing those ligands will move inside a cell, right? Molecules bind to receptor proteins on the membrane surface. So here they're binding, triggering vesicle formation. An example would be movement cholesterol and ions inside a cell. This is pinocytosis. Pinocytosis is a movement of fluids or small molecules into a cell. The small vesicle will be formed, pretty much membrane will just form this indentation inside, and then 2 ends fuse together, and some water and dissolved substances just move inside the cell. So this is often referred as cell drinking. This is the example of the phagocytosis. So we have pinocytosis and we have phagocytosis. Now, in the phagocytosis, the cell will form extension of the cytoplasm that is called pseudopods. So pseudopodium is like a feed. So they form this feed, and podium means food. So they form this extension of the cytoplasm. They surround the particles, and those are larger, bigger particles like bacteria, viruses, cellular debris, other foreign materials. And this particle, whatever it is taken inside a cell within those vesicles that is called phagosome. All right, and so yeah, and so this very often referred as cell eating. So again, endocytosis is active transport. It's movement of substances inside a cell. And of course, you always use ATP to do it. And can be different types of endocytosis: receptor-mediated endocytosis, pinocytosis, and phagocytosis. We also move substances out of a cell, and we call it exocytosis. And it can be constitutive or regulated secretion. Now, if you look at this diagram, what's shown here is Golgi apparatus, that is an organelle within a eukaryotic cell, and vesicle can be formed from Golgi apparatus, and what is inside, it might be some newly synthesized proteins. And then this vesicle moves towards the cell membrane. Right there, it fuses with the membrane and release substances or secrete substances out of a cell. Some secretion is regulated, like a release of calcium, for example. You don't want cells constantly secrete calcium into extracellular fluids, so you need some signal, right? So when this signal is received, the signal will be transmitted inside a cell, and this signal will stimulate formation of the vesicles. And again, vesicle will move towards the membrane and release substances or secrete substances outside a cell, right? So that's exocytosis. right. So I just want to kind of get back and like see what we covered so far. So we talk about cell theory, that cells are living units of life. We also said that cells are surrounded by extracellular material. We look at generalized cell, and we pretty much started to talk about cell membrane first, because every single cell has a cell membrane. We talk about structure of cell membrane. We talk about proteins, membrane proteins and their functions. Right, we mentioned glycocalyx and how cancerous cells can change their glycocalyx, so now your immune system cannot recognize and destroy those cells. And because we're talking about cell membrane, well, the cell membrane surrounds entire cell and it gives it a shape and form, right? But it also allows substances to move into and out of a cell. And that's what we call transport. We either can have passive transport or active transport, and we cover different types of active and passive transport. And now we will briefly look at different organelles rather than cell membrane, because cell membrane considered to be an organelle as well. Now, within the cell, we have a nucleus, and between nucleus and cell membrane, we have cytoplasm that equals cytosol, that is the liquid portion, and organelles. Right, so here you can see just a diagram show you all the different organelles. And those are very brief function, like vacuole is storage, ribosome is site of protein synthesis, smooth ER involved in a lipid metabolism and detoxification of some toxins, rough ER involved in the protein synthesis, Golgi apparatus sorts and package proteins from rough ER. Now I'm going a little bit maybe too quick over this, because we will have a diagram when we look at these functions in more details. Now mitochondria produce ATP for cell, lysosomes digest foreign substances and worn out organelles. Centrioles help to form spindle when cells divide during mitosis. Nucleus holds DNA. Nucleolus is inside nucleus, that is site of ribosome synthesis. And we talk about cell membrane already. Right, so let's kind of return back now over here. So you guys need to be able to recognize that this is nucleus. Nucleus is surrounded by nuclear envelope. Inside the nucleus, you have DNA. And then you have like very condensed region inside nucleus that is nucleolus, shown over here. Now, continuous with a nuclear envelope. So over here, that's shown in blue, envelope, and it continues with this membranous organelles called ER. And this ER has these little dots, ribosomes attached, and we call it rough ER because it has this rough appearance. Now, those dots are also ribosomes, but they are free floating ribosomes. So ribosomes can be attached to ER, making it rough ER, or it can be just in the cytosol. Now, this is a smooth ER. It's also kind of continuous with the rough ER. The difference is smooth ER doesn't have ribosomes. And ribosomes are important for protein production. And you can imagine if ribosomes are attached to ER, that means this rough ER also involved in the protein synthesis. And a smooth ER, if we don't have ribosome, it cannot do much with protein synthesis. So smooth ER for lipid synthesis, for carbohydrate synthesis, for lipid metabolism. So you not only synthesize, you can break down lipids within this smooth ER. Now, this organelle here is Golgi apparatus. So when proteins are synthesized by ribosomes, they deposit it inside this rough ER, then some vesicles will form and they will move to Golgi apparatus. From Golgi apparatus can form lots of vesicles. Some of them can be even lysosomes, some will move towards the cell membrane and secrete stuff out. So that's what I told you. We will have diagram to explain it in more details. Now we have vacuoles. Those are storage containers. Peroxisomes, those the organelles that will break down some substances that are dangerous for a cell and make them not toxic, right? For detoxification of some molecules over here that can be dangerous to a cell. Now, lysosomes, they have digestive enzymes inside. So this is mitochondria. Mitochondria here. So mitochondria is plural, right? So it should be shown into at least two organelles. But mitochondria for synthesis of ATP. Centrosomes have centrioles, and they important in a cellular division. And then you have all these lines shown over here. Those are proteins that form cytoskeleton. Okay, so here's some homeostatic imbalances. Some people have a mutation that prevents them from synthesizing lysosomal enzymes, especially in people with Tay-Sachs disease. I would say kids with Tay-Sachs disease, because those kids unfortunately die at age 5. So very severe genetic condition when patient lacks a lysosomal enzyme needed to break down glycolipids in brain cells. Now, glycolipids, those are fat with sugar attached to it, right? So pretty much the brain became fatty. So if you see over here, I think that's MRI of a brain of a patient with Tay-Sachs disease, and this is normal brain. See how it's completely damaged. And because we cannot break down those glycolipids, they will build up. So they will build up, and it will interfere with nervous system. So the brain stop functioning. It causes seizures, mental retardation, blindness, and death before age of 5. Now, those digestive enzymes are inside lysosomes. And here's our lysosome shown over here in the pink color. So if somebody lack of this important enzyme for glycolipid breakdown, then they have this Tay-Sachs disease. So you can imagine how important every single organelle has extreme importance in the health, overall health of people and any, other living creatures. Now, cytoskeleton, we mentioned it a little bit. Those are proteins within a cell, and they provide support, allow for transport of materials, for movement. Either, cells can have like cilia, flagella. They can move something across a membrane, or cell itself can move. Also, those those proteins are important in the muscle contraction. And so you can see they can be different in diameter, from microtubules that are the largest in diameter, intermediate filaments, and microfilaments. So cells are not boring. Inside the cells, you have the whole pattern of different proteins that allow for stuff to move within the cell, give it shape, give it support, right? And some important properties such as muscle contraction, for example. Now, we also mentioned that in the nucleus we have DNA, all right? Now, so DNA, what is this? Yeah, so this is just show us nucleus on its own, kind of, so you can see there is some transcription factors that are proteins inside the nucleus. There is some RNA, but most of the space, there is also nucleoplasm that has, lots of water, but the large portion of nucleus is occupied by DNA. Now, DNA shown on this picture is nucleic acid. So you need to refer to chapter 2 if you want to review structure of nucleic acid. But DNA is this double helix molecule shown over here. So it's a helix of DNA. And DNA is really pretty huge molecule. And you know what happens if you have these thin and very long molecules, and if they're not organized, they will look like this, right? So those are so tangled strings or threads that it would be really difficult for DNA to carry on its functions. That's why DNA cannot be just thrown like this inside nucleus. Instead, DNA is organized, kind of like shown on this picture, right? It's analogy. When we have those strings in the pools, now the DNA is wrapped around histones, around the proteins called histones. And when we have this DNA and histones and a little bit RNA, we call it now chromatin. So chromatin is made of DNA, histones, and RNA. Now this is more condensed form, it's more organized, but it's still pretty loose, right? And this is how DNA exists within a cell for the 90% of a cell life. But at specific time when cell is ready to divide, this chromatin will condense and then it will form chromosome. So chromosome is condensed chromatin, and chromatin is DNA, histones, and RNA, right? So pretty much this is different structural organization of DNA or DNA packaging. And we also have nuclear envelope and nuclear pores, and those are openings that allow substances to move in and out of nucleus. The DNA, however, stays within nucleus all the time. Right? Only when cell divide, when this nuclear envelope is broken into pieces, right, and it's pretty much disappears, we don't have it, then obviously chromosomes are not within nucleus, just because we don't have nucleus. But as long as we have nucleus, DNA stays inside. RNA can move in and out, proteins can move in and out, some ions can move in and out, but not DNA. All right, so, and as I said over here, we're gonna look at one of the cellular function that is protein synthesis, and we will see how these organelles are involved. I would even start with nucleus, right? Because right there, this little yellow part, this is where nucleus would be. This is where we would have DNA, and DNA would have a gene, specific genes for protein synthesis. So one protein, one gene. So to synthesize different proteins, we need those genes or instructions how to synthesize proteins, and those genes are part of DNA. But anyway, when we're not going through process of transcription and translation, but when proteins are synthesized, and proteins are synthesized by ribosomes, so those dots are ribosomes, and they're attached to ER, making it rough ER. Those proteins, newly synthesized proteins, they are deposited in those cisterns, right, inside those, that's kind of cisterns or lumen of rough ER. Then this membrane of the rough ER, it pinches off this organelle, it's pinched off with the proteins, synthesized proteins inside. Right, so if you say those ribosomes make proteins, right, then what RAF-ER does? Well, obviously you can see that RAF-ER concentrates those proteins, right? Instead of having ribosome here making, you know, protein, ribosome here, now we can, you know, concentrate and accumulate those proteins. Plus, inside those cisterns, we can have enzymes that will modify these proteins. But those proteins are not ready yet to be secreted out of a cell. First, in those vesicles, they pinched off the RAF-ER, those vesicles will move towards Golgi apparatus. And within Golgi, you see vesicle fuses, proteins are released now inside the lumen of Golgi, and they're gonna kind of move through it, right? And in the Golgi, you have what you call the cis phase. Now this is cis phase, and this is forming phase. This is where transport vesicles arrives. Then proteins move within ER, and we have lots of enzymes there as well that will modify those proteins, and they will package those proteins in a different vesicles. Now we call them secretory vesicles on the trans surface or maturing surface. And now those vesicles will, you know, have the different destiny, depends, right? What can, you know, what type of protein is inside. Maybe some of this vesicle will move towards cell membrane, fuse with it, and secrete those protein by exocytosis. Or if we synthesize digestive enzymes, those vesicles can become a lysosome. And lysosome will fuse with another vesicle, phagosome, that contains maybe some bacteria over here, or it can be, you know, some nutrients. They can fuse, and those digestive enzymes will digest whatever this phagosome has. For example, it might be a bacteria or virus, or some vesicle can just move towards the cell membrane, and that's how we replace the membrane. We can incorporate those vesicles in. For example, when cell is growing, when cells are just formed, they are smaller. So if you want cell to be bigger, you know, we need to increase the surface of cell membrane, or this cell, you know, membrane can be, you know, not functioning already, so it's need to be replaced by a new portion, right? So you maintain cell membrane, you allow it to get bigger, increase the surface area, and so on. So you can see how those organelles work together, right? Like ribosomes, RAF-ER, Golgi, then vesicles that form from Golgi. And that's the example of protein synthesis. Right, so yeah, we do have some other example. I just don't want to make this video too long. It's already getting extremely long. Okay, well, but that's pretty much it for the first part of this chapter. So if you guys are in anatomy class, that's all you need to know, right? If you in physiology class, then I will record the second part of this video, and we will cover physiology of cells, right? So anyway, we cover the cellular level of organization. Thank you for watching, and I hope it was helpful.